Welcome everyone. Thanks for joining our webinar on islet beta cell dysfunction in type 1 diabetes. I'm Tal Murthy, Global Product Manager for Diabetes and Obesity Product Portfolio at Outco. The total webinar is going to be about 45 minutes, and I will be moderating this session today. We have a pretty well-known speaker today, Professor Raghu Mirmira. Most researchers working in islet biology and diabetes probably already know him or his work. However, I would like to formally introduce him. Dr. Raghu Mirmira received his bachelor's, MD, and PhD degrees from University of Chicago. He's currently a professor in pediatric diabetes and director of the Diabetes Center at Indiana University in Indianapolis. During his research fellowship program, he held the prestigious Physician Postdoctoral Fellowship Award from Howard Hughes Medical Institute, a JDRF Career Development Award, and later a Research Career Award from National Institutes of Health. He's also a physician, heads a team of research scientists, and as an associate editor of Molecular Endocrinology and Journal of Diabetes. Dr. Raghu has in-depth experience in transcription factors that direct the development and function of beta cells. His research has more recently focused on mechanisms by which inflammation affects the islet beta cell in type 1 diabetes. The title of his talk today is Islet beta cell dysfunction in type 1 diabetes. If you have any questions during the webinar, please use the chat room or the question box to submit the questions. All the questions will be answered at the end of the webinar. So without further ado, I will pass on the controls to Professor Raghu Mirmira so he can go on with the presentation.
Okay. All right. So thank you, Tal, for that nice introduction. And and I also appreciate the opportunity to speak today about an emerging area in islet biology research, specifically the role of the beta cell in type 1 diabetes. And this has been an area that over the past, I would say, 2 to 3 years has been receiving more and more attention. While we always traditionally thought of type 1 diabetes as a disorder of the immune system, I don't think that that's fundamentally changed. I think now there is a new perspective that it's really an important interplay between the beta cell and the immune system. And I hope today to provide some evidence, this is all based on the published literature, that the beta cell does undergo a certain level of dysfunction, and that dysfunction may in fact either trigger or exacerbate the immune system in type 1 diabetes. So as you can see in the first slide, the prevalence of diabetes in the United States has been increasing quite dramatically in recent years. And in particular, this was a study published a few years ago based on N. Haynes data from 2005-2006, which showed that the cumulative prevalence of diabetes is close to 40% in the U.S. population. And what you can see here is that as you look by age brackets, you could see that even in that 12 to 19 age group, the prevalence of diabetes is nearly 20%. In large part, this is accounted for by the increasing prevalence of type 2 diabetes. However, a pretty substantial fraction of this percentage prevalence is certainly accounted for by type 1 diabetes. In fact, if you look in age groups at type 1 versus type 2 diabetes, what you find is that when you look across all ethnic groups under the age of 10, you could see that type 1 diabetes is still by far and away the most prevalent form of diabetes in the young. In the 10 to 19 age group, so obviously older children, what you see is that the prevalence of type 1 diabetes decreases somewhat, and this is based largely on ethnicity. So you can see that the non-Hispanic blacks, Hispanics, Asian Pacific Islanders, and American Indians have a much higher prevalence of type 2 in that age group, whereas in the under 10 age group, it's still primarily type 1 diabetes. But suffice it to say, even if you look at that 10 to 19 age group, a pretty substantial fraction of diabetes in that age group is still type 1 diabetes. If you look at a variety of different studies, and this is a search study, indicated that the incidence of type 1 diabetes for unclear reasons has been increasing in the United States. And this is data from Colorado. And the underlying cause of this increase is not entirely clear, although several hypotheses have been proposed, including the accelerator hypothesis as well as several others. But again, the point here being that it's not just type 2 that's increasing in prevalence, it's also type 1 diabetes. If you think about the disorder of diabetes, I think everybody who's probably listening in is aware that type 2 diabetes is thought now to be not just a disorder of insulin sensitivity, but also a disorder of beta cell function. And we're beginning to sort of appreciate that in type 1 diabetes as well. And that in both forms of diabetes, maintenance of beta cell mass and function are probably critical. So this maintenance of mass is really a sort of a state of balance. We know that different forms of diabetes, as you can see, type 1 and type 2, can lead to beta cell destruction. And that needs to be balanced to some extent by neogenesis and replication. And what we know about humans and older mice is that the process of replication certainly, and quite possibly neogenesis, is dramatically impaired as you get older. And so as you get older, if you have more destructive processes, the ability to sort of maintain beta cell mass at that point decreases dramatically. And therefore, that's part of the reason why diabetes prevalence seems to increase with age as well. In type 1 diabetes, we think of the disorder primarily as a defect in beta cell mass. We know individuals who are genetically at risk experience some type of environmental trigger. Again, the specific trigger could be varied, but we don't entirely know what all the triggers are. And then at some point, you develop antibody positivity, and then there's a decline in beta cell mass. And we clinically diagnose diabetes when mass declines to about 10 to 20% of the starting mass. And arguably at that point, you have so little mass, and when you couple that with the lower replication rates that we see in humans, the disease is pretty far gone. But however, in that sort of insulitis and pre-diabetes phase, where mass is still, for the most part, preserved, we see clear pathology in the islets. And this is from the NOD mouse. You can see the image of the islet stained in brown and then these invading immune cells. That's not a typical picture that we see in humans, though we are, again, that depends on the individual that you look at. A lot of the studies now from the NPOD seem to suggest that the raging insulitis that we see in NOD mice doesn't quite occur in humans. So the immunology of type 1 diabetes, I'm not going to go through it here. This is just a summary. But it's thought that it's triggered by exposure of beta cell antigens, which then result in the eventual activation of T cells that then cause homing to the islet and then eventual destruction by CD8-mediated, CD8-positive T cells. Now, most of the therapies as a result of this sort of understanding of pathogenesis has been aimed primarily at the immune system. And I show you two different studies here. There's a study with the anti-CD3 and then a study with a B-cell agent, anti-CD20. With either of these agents and virtually every agent that has been tried that has been successful, you see a picture that looks similar to this. That is that there seems to be a preservation for a period of time of C-peptide or what we believe to be beta cell function, and then there's a subsequent decline after several months that parallels the decline that you would see if the individual didn't receive any kind of therapy at all. So this suggests that what the therapy seemed to do is just delay the disease and the progression of the disease. And then it raises the provocative question, is targeting the immune system enough? It's possible that by intervening so late in the disease and given the low replication rates of the beta cell, you just don't have enough beta cell replication to increase mass. But clearly, the beta cell continues to decline even in the presence of these agents later on, suggesting that perhaps there are intrinsic processes that get started in the beta cell that then, when they're not dealt with, continue to progress. So that's caused many people in the field to rethink therapies and to perhaps focus on the beta cell a little bit more. It's important to think about beta cell function in type 1 diabetes because studies such as the DCCT here that I'm showing suggest that even if you can preserve some beta cell function, even in the presence of poor diabetes control, you still benefit. And you benefit because there's a decreased risk of kidney disease, a decreased risk of hypoglycemia, and a decreased risk of eye disease. So preserving beta cell function is clearly very important in type 1 diabetes. There was an interesting study published by Ellie Ferranini's group and the DPT-1 study group. This is the Diabetes Prevention Trial 1, that looked at beta cell function and then followed these individuals to whether they progressed or not to type 1 diabetes. And what was interesting in this study, if you just focus on the closed circles for a moment with the solid lines, you'll see that the non-progressors had sort of very robust beta cell function. And so what we're looking at here is insulin release in response to different varying levels of plasma glucose. So you can see insulin release is quite robust in individuals who never developed diabetes, whereas those who eventually progressed to diabetes had a somewhat flatter curve, even though they didn't have diabetes at the time. And this suggested that perhaps these individuals already had a signature of something going on in the beta cell years before they developed type 1 diabetes. And then, of course, by the time they develop diabetes, at the time of diagnosis, that curve becomes quite flat, consistent with the fact that they've lost substantial beta cell mass and function. So I'm going to just skip this. For the most part, I think everyone is familiar with the beta cell, which is a professional insulin-secreting cell. And it must effectively sense extracellular glucose and couple that to insulin secretion. And so when we talk about dysfunction, we talk about primarily the components that couple glucose sensing to insulin secretion being impaired in the cell. Okay, so we and other groups have studied beta cell dysfunction in type 1 diabetes, initially using the NOD mouse model. So I showed you some human data a few minutes ago. What do we see in the NOD mouse? And the NOD mouse is a mouse model of type 1 diabetes that displays autoimmunity. And as you can see here in our vivarium, about 80% of these mice get diabetes by about 20 weeks of age. And if you look in the pre-diabetic phase, when they're 6, 8, 10 weeks old, and you do glucose tolerance tests, you can see that here, the open squares are the NOD mice. Even when they don't have diabetes, they display impaired glucose tolerance. And that impaired glucose tolerance is coincident with a slightly diminished insulin secretion in response to a glucose load. And as you can see, as they age, even though they don't have diabetes, their insulin secretion tends to fall more precipitously with age. Interestingly, this isn't entirely a immune phenomenon, because if you look at the NOD skid mice, which doesn't have an immune system, even they show slightly impaired insulin secretion and worsening insulin secretion with age. So there's clearly something inherent about this line of mice on the NOD background. And so we've looked at whether this impairment in insulin secretion was due to mass or function. And so there's two possibilities. If you have reduced mass as a result of cellular death, you have less insulin secretion. But if you don't have reduced mass but just reduced function, you would also have impaired insulin secretion. So what we did was we, along with other labs, developed a PCR assay to detect beta cell death. I won't go through the details of this assay, but effectively it's looking at a specific epigenetic change on the insulin gene. And as beta cells die, they release their insulin DNA along with their epigenetic mark. And then using the serum from these animals or humans, we can detect the rare events that represent beta cell death just from the serum. And when we do that, here we show the NOD mouse, you can see that the beta cell death rate is higher in the NOD mice, and it increases slightly by 8 weeks and then begins to decrease by 10 weeks. And this is at a period of time when the animals don't have diabetes at all. So clearly beta cells are dying during this period of time. But if you look at the NOD skid mouse, which has no immune system, you can see that its level of beta cell death is also elevated compared to a background insulin diabetes resistant strain. So clearly there's death occurring in these beta cells. But is that the reason why they have dysfunction? It doesn't appear to be, because if you look at total mass in these animals, and that's in the black bars here, you can see the NOD animals, their beta cell mass is actually not changed. It doesn't change between 6 weeks and 10 weeks, even up to 13 weeks before they develop diabetes. So it's not an issue of mass. It appears to be that these beta cells are able to replicate at higher rates to maintain mass. So there's clearly a functional defect that's occurring at this very early age before they develop diabetes. And this is another study that was recently published by Rohit Kulkarni's group that showed the same thing. That is that beta cells in the NOD mice appear to have a higher rates of replication during the pre-diabetic phase. Other studies have shown this as well. This was a nice study published by Islet and Clayton Matthews a number of years ago, showing also that there is a defect in the early phase of insulin secretion in the NOD mice. So clearly there are defects in these animals of insulin secretion without dramatic changes in mass. So what's accounting for this? So one of the studies that we did a couple of years ago was we looked at insulin levels in these animals, and as I show you here, insulin levels seem to decline with age in the pre-diabetic phase. But interestingly, the pro-insulin levels actually increase. So they don't decline, but they increase. And if you look at the pro-insulin to insulin ratio, you can see that that ratio actually increases dramatically as these animals approach diabetes. And I think last month Bruce Verschere gave a webinar on the pro-insulin C-peptide or pro-insulin insulin ratio, and that there's clearly something deranged in these animals. So what would be accounting for this? So one of the things we've been interested in studying is this concept called endoplasmic reticulum stress. And when beta cells are exposed to inflammation, systemic inflammation that might be occurring from some kind of an environmental insult, the beta cells experience this inflammation and undergo, their endoplasmic reticulum undergoes stress, probably as a result of unfolded proteins or excessive oxidative stress. And as these endoplasmic reticulum expands, there is a, as you can see, protein misfolding, increase in proinsulin levels, and dysglycemia that then is compensated by a response called the unfolded protein response. And this response blocks new proteins from being produced and then attempts to fold those proteins that are misfolded. When this response fails, the next thing that happens is that you get endoplasmic reticulum fragmentation, and eventually when that happens, you don't produce protein sufficiently, you get cellular apoptosis, and then frank hyperglycemia. So that's sort of the end point. The question is this happening in the NOD mice? And again, as you can see here, these were islets isolated from the pre-diabetic NOD mice, and you can see all the markers of endoplasmic reticulum stress appear to be increased in these animals, including BIP, XBP1, spliced XBP1, and CHOP. But interestingly, some of the factors that are necessary to compensate for endoplasmic reticulum stress, such as ATF4, don't increase, suggesting that there's a failure to compensate for these animals. And then if you look at their endoplasmic reticulum, you can see here is the skin animal, which has nice stacked ER next to the nuclear membrane. The NOD animals it's very hard to find those. In fact, what you see are very swollen ER that's fragmented and not nice and stacked. So there's clearly a defect in the endoplasmic reticulum of these animals. And then in human studies, this is published by Dysfunction group from Belgium using NPOD samples. He showed as well that markers of increased ER stress occur in humans with early type 1 diabetes as well, including markers such as CHOP, BIP, and XBP1. So this clearly happens, and if you were to prevent that, can you ultimately prevent diabetes? Well, this was a nice study published by Gökhan Pathogenesis group in Science Translational Medicine last fall, where he treated NOD animals with a chemical chaperone that allows protein folding, TUDCA. It's a bile acid derivative, and what you notice is that when he treats these animals in the pre-diabetic phase with TUDCA, he can almost completely prevent the occurrence of diabetes in these animals. And the other thing he noticed was that in addition to preventing diabetes, there was also a lower rate of islet invasion by the immune system. So this suggested that targeting the beta cell early in its endoplasmic reticulum stress could in fact reduce the incidence of type 1 diabetes. If you then look at all the genes that give you susceptibility to type 1 diabetes, there's about 40 of them or so, candidate genes. And this was a nice study, again, published by Dysfunction in PLOS Genetics a couple of years ago. What he observed was that when human beta cells are treated with cytokines, about 60% of the type 1 diabetes susceptibility genes are in fact expressed in beta cells. So these aren't necessarily just immune cells or the fact that the immune cells are the ones that are deranged to begin with. It appears the beta cells in the disease can also begin to express these susceptibility genes. So where all of these data lead to is the concept that perhaps the beta cell itself may be a trigger of autoimmunity. And this is a model. This is, in fact, a paper that we just published very recently in the journal Islet that suggests that some environmental trigger, which we have yet to identify, it could be, in fact, a viral initiator or something else, perhaps the gut microbiome, leads to inflammation in the beta cell. And that with prolonged inflammation, you can get activation of the unfolded protein response. And then if this proceeds to the point where many of these proteins that are misfolded get released, as misfolded proteins, they may themselves represent neoantigens that could actually trigger the immune system in the susceptible individual. And as a result, you can get raging autoimmunity. And the concept here is that if you can treat early enough these individuals, so if you can identify these individuals at an early stage and then perhaps treat the beta cell or the derangements in the beta cell, perhaps you can prevent the release of these neoantigens and perhaps mitigate autoimmunity or at least reduce the incidence of it in these individuals. So again, this is purely a hypothesis, but it is based on data, certainly in mouse models, that suggest that it is a potential avenue for therapeutics as we move forward. So I just leave you with this last picture, and that's the beta cell-centric model of type 1 diabetes, is that the beta cell in many ways, the islet itself, may in fact be in and of itself the central feature of type 1 diabetes, because certainly the beta cell has the antigens that trigger autoimmunity, but it's possible in fact that we don't have to think of this loop that involves regional lymph nodes and so forth, that antigen presentation may actually be occurring in the vicinity of the islet, that T-cell polarization, formation of very angry CD8 T-cells might be occurring also in the vicinity of the islet, and that everything might actually be sort of centralized in the islet, not having to involve the entire immune system loop. And bringing again the question of whether therapies in the future should either focus on the beta cells or at least have a component that is a beta cell focus. So I'm going to stop there. My 25 minutes is up. And I just want to acknowledge the people that I collaborate with around the country, the people here in my lab who have contributed to some of the work that we've done, as well as my collaborators around the country, Jerry Nadler at Eastern Virginia, DCO Zurich in Belgium, and others, as you can see here as well. So I would be happy to answer any questions at this point.
Professor Raghu, thank you for the excellent talk, and thanks to all the attendees who are present. Just before going into the questions and just a couple of slides, I want to briefly mention that the ALPCO provides ELISA kits for assessing the beta cell health. This slide basically shows our latest chemiluminescent assays for beta cell health. We have a lot of scientific literature on these assays. If you have any questions, please visit the website or contact me directly. We also have a broad range of colorimetric ELISAs. Again, if you're interested, you can find a lot of information about these assays on our website on www.alpco.com. Again, at any time, if you want to reach out to us via email or phone, you can always get this information about the assays. So without further ado, I'll go into the questions. And Raghu, you can take the question. Which comes first, beta cell death or release of antigens?
That's a good question. So I don't know that we know the answer to that question entirely, but I would argue that it's probably release of antigens. This is, again, a controversial area. There was an interesting study published in the Journal of Immunology, I believe, a couple of years ago that overexpressed anti-apoptotic proteins in NOD mice, beta cells. And what they showed was that even though they were able to suppress beta cell death with those anti-apoptotic proteins, I think it was BCLX and a couple of others, what they observed was that it did not decrease the frequency of autoimmunity and eventual type 1 diabetes. And so this certainly suggests the possibility that perhaps when we think of beta cell death, it's probably really not necessarily the culprit, but that it may be dysfunction of the beta cell and then abnormal release of these antigens that may trigger autoimmunity.
Thank you. So next question, Raghu, this is for you again. Proinsulin-insulin ratio has been proposed to have beta cell dysfunction predictive value. Do you see that in your mouse studies as well?
Yeah, we do. And as I showed you in that one slide earlier, when we looked at the proinsulin-insulin ratio in mice, that we observed an early increase, in fact, weeks before, almost 6 weeks before these mice developed type 1 diabetes. So I think that's true. And there have been several human studies using older assays that suggested that in individuals who have antibody positivity, that there is an increased proinsulin C-peptide ratio. Of course, in humans we use typically C-peptide. So I think it's true in type 2 diabetes. I think it's true in type 1.
Thank you. This question is probably for me. Do you have a kit that measures intact proinsulin levels? Yes, we do have a chemiluminescent proinsulin kit. You can find a lot of information about that on our website. Next question, Raghu, again, this is for you. Does chaperone protein exposure retain ER histological structure?
Yeah, so that's a good question. I don't know if, so we have not done that study by giving, I assume that by that question you're referring to providing either small molecule protein chaperones or even the transgenic mouse model overexpressing protein chaperones. In that study by Hado Mislagil's group, I don't think that they looked at electron micrographs to actually look at ER morphology. We assume that it does based on the functional readouts that we have. We have, improved proinsulin levels, and then evidence that insulin secretion is restored. So we've never looked.
Great. We have another question. What are the differences between mice and humans in changes in beta cell function as type 1 diabetes mellitus progresses?
Yeah, that's a very, very good question. So, what we're trying to do right now in the field with human studies is focus more on prevention. So trying to understand better the pre-diabetic phase of type 1 diabetes in humans and whether that really truly does mimic what we see in mice. And I think because of this, there have been a lot of initiatives both from the JDRF and the NIH to focus more on this pre-diabetic phase. And the TrialNet network that does a lot of these studies have in fact moved to primarily focusing on diabetes prevention. And I think a lot of that will depend on really understanding the natural history of beta cell function. So I would say that we just don't have enough information from humans yet to say whether it is or is not similar to mice. We know that both exhibit beta cell dysfunction. The question is, do they exhibit the dysfunction for the same reasons? And I don't think we have that answer yet.
Thanks, Raghu. This question again is for me. Do you have a mouse proinsulin only kit? Yes, we do have that, and if you contact me directly, tal.murthy@opco.com, tmurthy@opco.com, I can actually provide that information. You can also get that information from our website. There's another question for you, Raghu. What's the evidence for beta cell stress dysfunction not being a result simply of increased metabolic demand in the context of partial loss of beta cells during the pre-diabetic period?
Wow, that's a complex question. So, we know that here's what is interesting. If you look at total beta cell mass of individuals with type 1 diabetes, in fact, total pancreatic mass in these individuals is actually smaller than in control individuals. And so one of the provocative questions this raises is, do people who are destined to develop type 1 diabetes have less mass to begin with? And as a result, does that alone put these beta cells at greater risk? And I think that's a great question. So if you have less mass to begin with, you knock off a few beta cells, you get a little hyperglycemic, that hyperglycemia comes back, causes oxidative stress in the beta cell, then you knock off more beta cells. Could that be the cause of this dysfunction early? And I think that that's a legitimate question. Do you really need inflammation? Do you really need that sort of, signaling cascade involving cytokines to trigger this? And I don't know if you do, but it certainly is a thought that perhaps just intrinsic stress from just not having enough beta cells and having a little bit of hyperglycemia could really trigger all of this.
That's great. Thanks, Raghu. We have one more question. How would you imagine a screen, proinsulin-insulin ratio, beta cell death, PCR-based measures yearly? How do you imagine a screen to be?
Oh, so how would a screen look? Okay. So I think what you're talking about is a biomarker type screen. So I think the people that have been working on this type of thing, what we call the Diabetes Prevention Trial Risk Score, the DPTRS, is really sort of what we're kind of driving at, and that is that there's probably going to be a variety of factors that figure into an equation. So it could be things such as just anthropometric data, such as body mass index and ethnicity and things like that that could be part of that equation. And then another component of that could be, for example, we could be looking at things like proinsulin levels, could be part of that equation. Antibodies, of course, could be part of that equation. So I think a screen would likely involve, to increase the ability to predict type 1 diabetes, would probably 5, 6, 7 different parameters that span everything from blood, measures.
It's for me. Will you offer slides for download? Yes, we will make the slides and the recorded version available in about a week's time. Another question, Raghu.
Mm-hmm.
Does the increase in proinsulin correlate with elevated beta cell death in pre-diabetic phase?
Well, yes, it does in our mouse studies, but whether it's, one is causing the other or whether they're occurring simultaneously, independently, we don't know the answer to that. So we've been looking, as I showed you in our data, when we looked at beta cell death increased in NOD mice at the same time proinsulin levels went up. So it's possible, and this is just my guess, that there are some dysfunctioning beta cells that release more proinsulin, and then at the same time there are some beta cells that are dying, and that you're seeing two different things occurring simultaneously. But whether one is causally linked to the other, I don't have any data on that, and I don't know that anybody really knows that question, or the answer to that question.
Another question, does the characterization of T-cell phenotypes have any value using flow cytometry in type 1?
So T-cell phenotypes, is that right? I think it does. In fact, we're beginning to appreciate the fact that the absence of regulatory T cells, these are the CD4, CD25, FOXP3 positive cells, correlate with the occurrence of type 1 diabetes. So it's possible that if you have more of the bad T cells, what we call the Th1 cells and the CD8 cells, and then less of the regulatory T cells, that is likely to put you at greater risk for developing type 1 diabetes, and quite possibly that could be another marker of the disease. The only question is that, what you measure in peripheral blood, is that reflective of what's actually happening in the pancreas itself or in the pancreatic lymph node? And so unless you have a robust and specific T-cell phenotype that you can go after, it could be pretty difficult. But I'm not an immunologist, so I don't know that I'm the right person to ask that question to. So I will point that caveat out.
Thanks, Raghu. There's one more question here. How can we use the NOD data in humans so far?
Yeah, as a matter of fact, the NOD data have not been, I think this is the way we've been using it. If it works in an NOD mouse and it makes sense, you might try it in a human. If it doesn't work in an NOD mouse, then you don't even waste your time in humans. And those that do work in the NOD mouse, by far and away, the majority of them, don't have the same robust effects that we see in humans. So, you can actually reverse diabetes in an NOD mouse. We have yet to reverse diabetes in a human. So I think The NOD mouse is a great model, but I don't think it is really where we are putting a lot of our efforts on going forward these days. And a classic example is the recent call from the NIH for the Human Islet Research Network grants, which are really focused almost entirely on humans and not on NOD mice. And I think that's probably a direction to go. We hear a lot as well about humanized mice. Again, my problem with humanized mice, many of these mice have human immune systems. I think it's really cool, but again, they don't have human beta cells. And so I think the caveat with any of those humanized mice is that you still don't have that other component that appears to be really important, and that is the human beta cell. So I think it's the best model we've got, but we've got a long ways to go.
One more question, Raghu. Are apoptotic mechanisms increased in beta cells in the pre-diabetic phase?
Yeah, yes, they are. You can see the activation of apoptotic pathways in these beta cells. We saw it ourselves. We saw evidence of caspase and CHOP activation in the pre-diabetic NOD mice. DCO Aziric looked in humans. Now, granted, they had type 1 diabetes, but they were very early in the stage of disease, and they did see evidence of increased apoptosis. So I think that apoptosis clearly does occur in that phase. Some people would also argue that quite possibly necrosis also occurs in that phase. So I think it does happen. How important that is in the pathophysiology of the disease, we don't know yet.
Thanks, Raghu. One more question. Is there any evidence that ER stress triggers beta cell death in type 2 diabetic patients or rodents?
Oh, wow. So some people would argue that ER stress in and of itself doesn't really trigger death. And again, a lot of it depends on, the literature you read and the mouse or the animal models that you look at. Some people would argue it primarily causes dysfunction but not frank death. Other people would argue that it causes death. What we don't really know right now is how much death there really is in type 2 diabetes, death of beta cells there is in type 2 diabetes. I think, again, there's new concepts being raised in this regard. For example, could ER stress be causing a phenotypic change in these cells so that they're not really beta cells anymore? And I think that's a legitimate question to ask. And how important in humans is the loss of beta cell mass in type 2 as opposed to beta cell function? I think these are really great questions that aren't entirely answered, because I think we have a pretty good sense in the mouse, but even there's controversy. But in the human, I think we have virtually no idea.
Thanks, Raghu. There are a few more questions, but I think we are running out of time. And I do have my contact information here. Tal Murthy, you can reach me anytime with any questions. And you can also contact Customer Solutions, cs@outco.com, for product-related information. Just want to remind you again that the slide recording and the slides will be available in the next few days, perhaps this week or early next week. Again, I'd like to thank you, Raghu, for the excellent talk. It was very informative. And thank you all for attending the webinar as well. Again, at any time, feel free to contact us. We are also very interested in the beta cell health, and we'll try to accommodate your request as much as possible. Thanks again for attending, and have a good day. Thank you.
Thanks. Bye.